Okay, yes. Others they drop off, right? They go home, they want to be here. That's great. In other words, this is a tough business, and it's a hard problem. Congratulations for those who are here, and you guys are great. It makes me feel better. At least I'm talking to a person and talking to people instead of all chairs. Anyway, today I just literally my last lecture in my series. Can you hear me? I believe it's easy. Just one. So I want to talk about something about cancer. The reason I'm talking about cancer is, as a dentist, eventually you will deal with your patients, like aged patients, the people with different complications in their oral cavities. Cancer obviously is one of the complications you may encounter in your future practice. So that's why I want to talk about cancer specifically. You have better understanding of cancer, then eventually you may need to explain to your patients what our cancer is and so smoking is bad. Everybody knows smoking is bad. Smoking is associated with oral cancer in oral cavity. But why is that? So you really need to know what's going on here. And also you may deal with patients who have different cancers in the other parts of the body, like lung cancer, breast cancer, and so on. And maybe you have some patients under different cancer treatment, and then if you have better understanding about the things I'm talking about, I'm talking to give you as like a physician to treat a patient, but however, it can be a tool for you to become a better communicator with your patients. That's the purpose. I mean, definitely understanding the basic science about our body is the main purpose of this lecture. So in this lecture, I'm going to talk about some cancer biology, cancer-associated immunity, and cancer immunotherapy. So the reason I'm talking about focus on, and also mention about cancer immunotherapy, just because this is a red hot topic these days, and it's pretty much the last hope for cancer treatment. And the first thing, so again, I mentioned about I want to focus on cancer biology instead of like histology, diagnosis, or treatment, those kind of things, just you have better understanding what makes a cancer cell, and also what's a cancer immunotherapy. Those are very basic things. And so I have listed some references. Again, I believe I couldn't upload it. The file is huge. I believe you already received a PDF format. Mm-hmm. And did you? I don't think please see. Can you read the notes? I mean, for some of those slides, I put the notes in the notes section. Okay. Yeah, I'll try to send out a link so the course director can directly upload the file from the link instead of my email. Email didn't work. I couldn't make it work. So that's why I have to compress it, make it smaller. And so today actually for me it's really heavy, and a lot of things I'm going to talk about the other thing. So I heard from other instructors talking about videotaping, those kind of things. You guys still want to do it? Yeah, if you want to do it, then just do it. I don't care. I mean, I would like you guys to videotaping it so you save time instead of still focus on the lecture. I'm talking about what I'm talking about, but you have some feeling, you feel safety afterwards, okay, to prepare for exams. And so I just want to talk about some general categories of cancers, and then specifically several minutes on head and neck squamous cell carcinoma, HNSCC for short. And then I mentioned about tumor suppressor genes and oncogenes. And then I'll talk about, because cancer is something that those cell growth is out of control, and then there is something at the molecular level associated with that, it's called cell cycles. And then I'll mention about immunotherapy. So it's up to how much the course is progressing. We will determine whether or not we won't have we won't have a break. So the cancer is the second most common cause of death for any diseases in the U.S. Which one is the first? Heart diseases. So right now, so heart disease, the death caused by heart diseases is getting lower and lower. However, cancer definitely has been reduced, like in this country, by 13% during the past 10 years. And so, again, this still is the growing killer for human beings. So that's why there are a lot of energy from the federal government, from companies, and from local governments and individuals to put in the cancer research. So then what's cancer? Cancer is not a single disease. This is, we have to be sure. So actually, this is a vast variety of disease involving abnormal cell growth, not normal cell growth. Normal cell growth is happening all the time. That's good. And with potential to invade or spread to other parts of the body. So this is literally the definition of cancer. And cancer can occur in any organ system through the animals. So not a human will have cancer. Dogs, cats, they also have cancer. And this is a chronic disease. It's caused by defective genome surveillance and the signal transduction mechanisms. Because the signal transduction related to the cell growth and proliferation or migration become dysfunctional, so out of control. Those are the put them together are the definition for cancer. So you will hear a lot of terms related to the cancer, like a neoplasm. So neoplasm basically is a cell proliferation. So those cell growth out of control. So basically, the neoplasm can be benign. In other words, those are normal cells. You get more normal cells, they form a tumor like a fat tumor. So you just remove it. Okay. The reason is they are normal cells, and they can malignant. In other words, they are not normal cells. So in this case, neoplasm is a cancer. And the other words for the characterization of cancer cells is anaplasia. This is a loss of the ability to differentiate. So in our body, the differentiation is good because those cells, they will have specific function and they can work. For example, like epithelial cells in oral mucosa, in our oral cavity and skin, they have their function as a barrier between the body and environment to protect ourselves from outside insults. So those are differentiated cells. Other examples will be heart cells, myeloid cells, and neurons. So those are differentiation. But if epithelial cells, they lost the ability to become epithelial cells, become like a mucosa, so they keep their original stem cell-like status. They have higher ability to become cancer cells. This process is called anaplasia. Transformation. So most of the time, so those cells they will gain self-sufficiency in growth cycle. So in other words, you don't have to give them those cells a growth factor. They can still grow. They are happy to grow because they can generate those growth factors, or they have mutation in the growth factor receptors to activate themselves, and including malignant transformation. Those are the terms we are talking about. The process from a normal cell becomes a cancer cell. So here I just give you some examples of cancer categories. One is carcinoma. So carcinoma is a cancer related to epithelial cells, no matter what, where the epithelial cells are. At least 80% of all cancers are carcinomas. So adenocarcinoma are cancers that starts in granular tissues, as in the ducts of a lobulus of breast, colorectal cancer, and some types of lung cancer. So I just give you a Chinese standing of this, how does adenocarcinoma cancer at the molecular level, at the cellular level, tissue level looks like. Those are cancer cells, and some of them already invaded into surrounding region. So you can still see the granular structure of those cancer cells, but some of them, they get out, become invasive cancer. So this is a squamous cell carcinoma. Those are the cancers that begin in the lung granular cells, for example, the skin or the oral mucosa. So oral cancers belong to the squamous cell carcinoma category. And this is the one example. This is a typical, it's called a fully differentiated oral squamous cell carcinoma sample. And this is from my lab. So we found, you can see the cell, this is the whole piece of the picture shown here. This piece, they are cancer. So, however, in the middle, in the center, or in the core region of the cancer, those cancer cells, they are difficult to get nutrients or blood. So, since they don't have enough oxygens and they don't have nutrients, they don't have any blood supply, they will die, undergo necrosis. They become like dead cells. And this, because those are epithelial cells, when they die, they will produce keratins. So this is called a keratin pearl. Whenever you see the structure of this is a very clear indication of a cancer. In my lab, we are working on different things. We just a standard tissue with two proteins. One is called HBD2, the other is called HBD3. So you can see that HBD3 is associated with cancer cells. This is the region called invasive fringe. In other words, those cancer cells can come out and spread to the other part of the organ. For example, like a lymph nodes. HBD2 is associated with the cancer cells, very close to the dead cell. This region full of necrotic cancer cells. So basal cell carcinoma, most of the time we will refer to lung melanoma skin cancer. This is because this cancer is never spread, and it's even. I saw many people, they have this basal cell carcinoma. It's treatable. They just remove it surgically, and they it's gone. Melanoma is also the cancer, so it's different from all the other carcinoma we are talking about, because those are the tumor that begins in the cells. It's called a melanocytes. Those are the cells produce skin color. I mean, this is curable in its early stages, but it can spread very quickly to the other part of the body if treatment is delayed. And sarcoma, specifically, we are talking about malignant tumors growing connective tissues like cartilage, fat, muscle, or bone. So the head and neck cancer happens in the head and neck region. And for example, like oral cavity, like tongue, hard palate, and the base of the tongue. So they have different, so in these two different regions, they have different causal factors. In the oral cavity, the cancers in the oral cavity is most likely related to tobacco use and heavy drinking. However, cancers in the oropharynx region, like tonsil oropharynx, base of the tongue, those are associated with human papillomavirus infection, HPV infection. So HPV virus does infect human in the throat. And this, however, this only takes about 15 to 20% of all head and neck cancer. And right now there is a motion from those big pharma companies to vaccinate teenagers with anti-HPV vaccination and try to reduce at least 20% of the oral cancer, head and neck cancer associated with HPV infection. So this is, I just give you a very brief statistics. Every year we will have about 50,000 adults in this country. They will be diagnosed with head and neck cancer. And the men will have higher incidence rate compared to women patients. The average age of the diagnosis is about 60 years old, so over 60 years old, it's kind of like a risk. And every year we will have about 10 deaths in the United States. This is constant for the several years. It hasn't been improved by the during the past 2 years. So oral cancer is really and the formation of oral cancer is a really complicated process. And it becomes from normal oral epithelium. You can see that this is a lower lip of the structure. And you can see those are the basal layer cells. And these are the cancer cells. These are the stem cells to form those basal layer cells. Up those basal layer cells, those epithelial cells more or less matured or differentiated epithelial cells. And once those patient has leukoplakia, so in other words, it's a white patch, this kind of lesion, and you can see a thickening of these basal layer cells, and they are still normal, even they gain the ability to proliferate. And further, some of those leukoplakia lesions, maybe like 0.5% of them, will become carcinoma incisal lesions. So in this case, all the cancer cells. So if you look at this figure, I really appreciate all the cancer. Those are the precancerous cells. They come out from basal layer, basal like stem cells. So that's I mentioned about the stem cells in specifically in oral cancer. So are the original source for cancer. And in my work, we found out all those cancer cells to only produce previous cancer, precancerous cells, they only produce a molecule called HBD3. And once they become a full-blown cancer, so those cancer cells in the closed keratin pearl, those are dead cancer cells. They produce beta-depensin 2, HBD2. Cancer cells invasion front, they only produce HBD3. So based on this work, we spent a lot of time to figure out why those oral cancer, they differentially produce HBD2 and HBD3, and why those cancer cells, like the stem-like cancer cells, as well as cancer cells at the invasion front, specifically produce beta-catenin 3. What's the function of cancer cell derived HBD3 in cancer progression? So this work has been received funding from NIH. We are spending almost $1 million in 5 years period to study this, to work on this. Really, we have some interesting findings there. And so, as I said, we are talking about basic sciences instead of the research. So again, what makes a cancer cell a cancer cell? Just because there are two groups of, yeah.
Can you go back a slide there? So it's a cancer, not a leukoplegia. It's not a cancer yet.
Right, leukoplegia is just a white, so white patch, white lesion. It can cause by any reason. So
Okay.
this is a clinical term.
So carcinoma in situ is cancer, it just hasn't broken through the basement membrane.
No, they don't grow. Right. So,
But it is a cancer at that point.
so once they, just like for example, we know those are previous. So those are cancer cells, and you don't. So they are retained by the basement membrane. So they are called like a carcinoma in situ. This is a typical carcinoma in situ lesion. And if you identify carcinoma in situ lesions, you just cut it, that they no longer become cancer. But if you remain this kind of lesions untreated, eventually they will become cancer.
So, but it does count as cancer at that third stage.
In this case, this is, for me, I'll count it as cancer. But
Okay.
for a clinician, say like Dr. Hirsch, he just retired, and he is an oral pathologist. He won't count this as cancer because the response to treatment is the prognosis is very good. And you cut it, literally they're gone. So the reason that a cancer cell becomes cancer, literally there are two types of forces in the cells. One is called oncogenes, the other is called tumor suppressor genes. So when I'm talking about the genes, I'm not talking about like genes in the chromosome. I'm talking about the genes and those the proteins encoded by those genes. And so many of those oncogenes were identified in viral infected like birds. So they found out a tumor in a bird and identified the virus. Those virus, they sequenced the virus. They found out, oh, there was in human cells, we also have the exactly same genes. And then they figured out, yeah, those cancer patients, they have the gene exactly same activated, just like the genes in those virus in birds, so in chicken. So then eventually those genes are identified, are categorized as oncogenes because they can form, whenever you have cancer, you will see those oncogenes overexpressed. On the other hand, we also have some genes called tumor suppressor genes, and I'll put more details there. And proto-oncogenes definitely, and just because those genes, they have the potential to become an oncogene. So what are tumor suppressor genes? So tumor suppressor genes protect a cell from becoming a cancer cell. So make sure it controls cell growth. So the cell growth is under control. They will no longer go into the uncontrolled cell proliferation, and they can induce cell death or apoptosis. That's what the other more professional term. And often mutated in cancer cells. So this is a loss of function mutation. So the tumor suppressor gene doesn't have the function. And therefore, I will induce the two-hit hypothesis. So we all know in the human body, we have two chromosomes, one from father and the other from the mother. And they mixed. So when I may inherit a mutant tumor suppressor gene from my mom, so in other words, it's not functional. However, I will have a normal tumor suppressor gene from my father. So this normal tumor suppressor gene can produce the tumor suppressor protein and just function as normal. I'm okay. However, if I have a mutation from my mom in a tumor suppressor gene, the same time later on, I smoke a lot, for example, and then that causes a mutation in the two other tumor suppressor gene, supposed to be normal from my father. So then I'll have the same tumor suppressor gene, all mutated. So I need a really like a two hit on the tumor suppressor gene to be found to lose the function of the tumor suppressor gene. So then the cells without a tumor suppressor gene or the tumor suppressors, they will when they receive the environment or like growth factors, different things like a carcinogen stimulates, and then those cells will become cancer cells. The most famous tumor suppressor gene is called P53, and this is the mutation of this gene is involved in over 50% of different types of cancers. And the other one is called retinoblastoma protein, RB1 for short, and there are so many of them. I don't want to list all of them here. So one is for P53, from the work I analyzed for the oral cancer patients, if they have mutation in the P53, two alleles of the genes, they all get mutated. So this is a typical two-hit hypothesis. So in contrast, oncogenes are totally different. Oncogenes, they cause cancer. They stimulate cell growth. They are anti-apoptotic. So they help cells they keep cells from dying, by all different reasons, by nature or something. And they often mutated or highly expressed in cancer cells. So there is a single hit hypothesis regarding to oncogenes, because if I have one oncogene is totally non-functional, I have other oncogene get activated because the mutation overactivated, and then I can get cancer. Just one hit is enough to cause cancer. Both of those oncogenes are growth factors. For example, for some reason, I put EGF. So this is an epidermal growth factor and epidermal growth factor receptor. This axis is mutation, this axis, most of the time, it's overexpression of EGFR, that's epidermal growth factor receptor, is involved in 90% of oral cancers. So this is, in my lab, we always use EGFR to identify whether or not this is the oral cancer cell line. And we tried really hard to knock out, kill the EGFR receptor in cancer cells. The interesting thing is, once we kill this EGFR, knock out the EGFR gene in oral cancer cells, and in a sudden, all the oral cancer cells are dead. Because those oral cancer cells really need EGFR, the growth factor receptor, to maintain their growth and proliferation ability. Other oncogenes, including protein kinases, called a RAF kinase, SERC, GTPases, like RAS, and transcription factors, MYC. There's so many of them there. So I don't want you to memorize all of these kind of genes. Sometimes I know them just because I read a lot. And so, however, I really need you should know to understand what's the difference between tumor suppressor genes and oncogenes, and the two-hit and one-hit hypothesis. Those are something really easy to understand. In the exams, so my test question will be something like RAF is an oncogene, right? So overexpression or mutation cause the dysfunction is related to cancer, and so you need a two mutation or one mutation or one hit or two hit to cause cancer. So if you think this is an oncogene, and then you think about a single hit hypothesis, you just need one hit, and that's enough to cause cancer. There are so many causal factors for cancers, and one is genetics, and the other is because the random mutations in stem cells is associated with most of the cancer. This is like a theory, others don't believe it. And carcinogens, typical example is that smoking is associated with cancer. Viral infection is related to 20% of all cancers. 50% of those viral infection causing cancer is related to persistent infection by human papillomaviruses. So the HPV infection is related to over 90% of cervical cancer, and it's related to 20% of head and neck cancer. So HPV infection is a big concern, specifically for head and neck cancer in this country, because right now the HPV causes the cases of head and neck cancer caused by HPV already surpass the cases of our cervical cancer. That's a typical causal factor for HPV infection for the cervical cancer. So this is a really big concern. And also, epidemiology analysis already show HPV infection in oral cavity and in throat is on the rise in this country, in men, specifically men and boys. So here I have a question. I mean, it's very long. Just take a minute, read it. Just tell me true or false. The single piece? False. I think this is a false. You're right. I'm glad that what I'm saying is delivered. I'm happy about it. Again, so what makes a cancer cell a cancer cell? Just because most of the time, I believe, personally, I believe that's a changes to genes. So some people don't believe it, they believe environmental factors that makes other things, but I believe the genes, it's called mutations. Those mutations play a very important role in the development of cancer. And because mutations can cause a cell to make proteins that affect how it grows and divides into new cells. And certain mutations can cause cells to grow out of control. And this is a really bad scenario, and that can lead to cancer. So many gene mutations associated with controlling a process is called a cell cycle. And then what's cell cycle? The cell cycle, so we know a cell multiplies through division. So you have one single cell become two cells. Specifically for the cells in our in oral cavity, because constantly you have a lot of mucosal cell death, and then you have reshuffle of those dead cells, and then you have epithelial stem cells to grow more epithelial cells to replace those dead cells and make sure we have maintain the homeostasis of the mucosal region. So the cell, so therefore there is like a cell, those cells multiply in oral cavity all the time, constantly. Others will be like blood cells and skin cells, and many of them you can think about it. So, however, the cell division is tightly controlled. If it's out of control, and then it can be detrimental. So this is like a cell division is really complicated biochemical and morphological events. So if you think about all the events from one single cell to become two cells to daughter cells, and those are the events are called like referred as cell cycle. So I just want to play a video, and this is a very simple video. Just tell you how one single cell eventually become two cells, and there are so many. The cell cycle can be divided into different phases: interphase, prophase, all the way to the cytokinases. From one single cell to become two cells, and so in my slide, in my PowerPoint file, I have a very detailed description about the cell cycle there, and so I'll try to upload my PPT file there. And if you look at those cells, so it's like it from one single cell eventually become two identical cells. So this is all the biochemical and morphological events. What's happening there is called a cell cycle. It's easier for people to understand. So when we're talking about cell cycle, we always start from, there are two different types of cells. One is permanently differentiated cells. They call it permanent cells, like neurons and cardiomyocytes. So they are no longer divide, and they can grow, because like if you cut them out, like if you cut a neuron, then they can regrow, but they are not going to proliferate. So they are, in most cases, they don't have the ability to become cancer, because they are fully differentiated and they are permanent cells. And however, many of the cells, they are in a quiet situation. So those are the stable cells. For example, will be hepatocytes, like our skin cells and mucosal cells. And so in this cell, the whole cell cycle can be divided into different phases. One is interphase. So in this phase, cell undergoes a lot of RNA, DNA replication, RNA synthesis, and protein production. And also we have cytokinases. In this phase, one single cell will physically become two cells. And so the cell cycle, you have to finish all the biochemical processes during the previous cycle, be ready for the next cycle. And therefore, it's not reversible. It's stoppable, but it's not reversible. Once those cells become, after like a cytokinases, there is no way for them to become interface. So that's an important concept about a cycle. So the cell cycle is not reversible. And however, it's stoppable. The reason it stopped somewhere just because the cells need to check out whether or not if there was any errors made during the process. For example, we generally divide the cell cycle become like there are several phases called a G, the gap, so G is, so the gap phase is a G for short, gap 0, gap 1, and gap 1 S. So S phase is somewhere, is the time we have synthesis duplication of DNA, and followed by gap 2. So here, cells will check whether or not our cell made any mistakes in DNA replication. For example, if there is a mutation, then cells will detect it and just simply kill the cells, right? Because we don't have any mutation. Mutations happen in the cell cycle. And then after G2 phase, if everything's okay, and then the cell will start mitosis. One single cells physically become 2 cells. And so this is the whole cell division. From 1 cell becomes 2 cells. So you just know the gap phase of G1, G2 are where the cells will stop. Okay? And to check everything, whether or not everything's fine. And so specifically the G1S checkpoint, it's called a checkpoint. It's important here. So you have a lot of RNA protein synthesis in this phase. And then just before the synthesis of DNA, so make sure you have enough energy for the DNA production. And also you need to make sure all the proteins are ready. So once those after the interphase, the DNA become duplicated. And you have synthesized the DNA, and then into the G2 phase, make sure all the DNA faithfully duplicated without any mutations. So if there is a mutation in the DNA replication, so the protein inside the cells will find it out, and eventually just simply kill the cells. So here is where the cancer cell, the normal cell becomes a cancer cell. So if the tumor suppressor genes cannot recognize a mutation in this phase, so the mutation will be carried on to the daughter cells, and eventually there is a possibility those mutant cells, the cells with mutant genes, can become cancer cells. So there are so many proteins that can modulate the cell cycle, and so most time the cell cycle is controlled by nuclear proteins that start, continue, and check for errors during the cell cycle. So you can think about it, there are so many proteins are involved, and just make sure the cell cycle is carried out correctly. And what are cell cycles? There are 2 groups of families of proteins involved in controlling cell cycle. One is called cyclins. So cyclins are a family of nuclear proteins involved in the progression of cell cycle. But those cyclins, they have no enzyme activity. So they are nuclear proteins, but they don't have enzyme activity. That's an important concept. Try to remember it. Because those cyclins can form complex with a different protein called cyclin-dependent kinases to activate the cyclin-dependent kinases to be functional during the cell cycle. So cyclins, they don't have enzyme activities. And there are several different types of cyclins activate at different parts of the cell cycle. I'll talk about that later. Then I mentioned about the cyclins can interact with cyclin-dependent kinases, CDKs for short. Those are key regulators of cell cycle. So CDKs are a large family of proteins. Those are serine-threonine protein kinases. So they have catalytic subunits. Cyclin-dependent kinases have enzyme activity. cyclins, they don't have enzyme activity. However, CDKs, they do have enzyme activity. They are able to phosphorylate other proteins, downstream proteins. However, activation of CDKs requires binding of cyclins, like a different cyclins bind to different CDKs in the cell cycle, and so that will make the progression of cell cycle smoothly without any errors. So if we look at it, this is the concentration of different cyclins and CDKs during a cell cycle. In the G1 phase, we have expression or production of cyclin D. Then cyclin D can interact with CDK6 to activate CDK6. Then CDK6 will activate, induce a phosphorylation of a tumor suppressor gene. So to inactivate the tumor suppressor gene, then cell cycle can go. There are many other things that can show the G1 phase. So for example, like P53 activates other tumor suppressor genes to inhibit CDK2 interaction with the CDKE, or they can directly inhibit cyclin D's CDK6 interaction. And growth factors are able to stimulate the production of cells, cyclin D, so to push cells in the quiescent state into the growth state. And followed during the S phase, so I mentioned about in S phase is DNA synthesis phase. So we have cyclin E production, and then that will activate CDK2, a different CDK cyclin-independent kinase, and followed by cyclin A expression, cyclin B expression. Together, they interact. They activate different types of CDKs to push the cell cycle forward. Yeah.
And then all of this is under the direction of the nucleus. The immune
Yes.
system is not involved. There's no immune cell watching over this in any way.
No.
Okay.
No, that's the point. So, say immune cells, if they want to divide in their nucleus, they will have same process going on.
But there's no external.
No external.
Check to
The
make
only
sure this doesn't
external
go out of
is
control.
here. You have, say, like if you have growth
Okay.
factors, and then that will stimulate cyclin D. For example, in the oral epithelium, I mentioned about we have those stem cells in the basal layer. So most of those basal layer cells are stem cells. So without these growth factors, they just quiescent, they don't grow. But once they have any growth factors produced from others, and then they will start proliferate
So
to get more epithelial cells.
either due to one hit or two hit, at some point, a cell becomes cancerous and starts over-dividing.
Mhm.
At what point does it get the attention of the immune system?
Yeah, so the immune system is a little bit different. I'll mention about that later.
Okay.
And so the immune system is something, so think about like this. The mutant cells, so all the cells in our body, our immune system don't recognize, they are not foreign.
Right.
So they don't do anything. The immune system just like regularly check if there was any, say, like infection or pathology in our body. You can consider that as a surveillance system, make sure everything's okay. And so for the cancer biology, we're thinking the cancer cells I mentioned about it, they are abnormal cells. So they are foreign, supposed to be foreign. And our system supposed to be able to recognize them, to kill them. However, our immune system didn't do it. It doesn't, so the reason they don't do the work just because they don't recognize those cancer cells as for just like part of our body. And for cancer cells, they have different ways, I will mention about that, to fool the immune system instead of to direct the immune system to the wrong direction, and to make sure the immune system couldn't recognize that. So the question will be the cells. Yeah, I think I finished it. So just one point to answer your question. So in the one hit hypothesis or single hit hypothesis, so growth factors can activate this to initiate the cell cycle, right? And they can it can make this cell growth out of control in hit, once or single hit hypothesis and a two hit hypothesis. Like most of the tumor suppressor genes requires mutation in two copies of the genes. So here the cell cycle is reversible. It's not irreversible, right? Cyclins have enzyme activities and stimulate cell cycle. Right, that's false. So I just want to summarize. So far, I'm talking about there are hallmarks of cancer. So far for us, we just need to know there are the tumor suppressor genes and there are oncogenes, and they work together to make a cell become normal cell, become a cancer cell. And also, I mentioned about cell cycle and cell cycle control. And cell cycle is non-reversible. It's controlled by cyclins and cyclin-dependent kinases. Cyclin-dependent kinases they have enzyme activity. However, cyclins, they don't have enzyme activity. Of course, I didn't mention about how those tumor suppressor genes are involved in the cell cycle and cell cycle control. And just because it's a little bit too complicated. It's impossible for me to explain to you in this really short lecture. I mentioned about the cyclins and the cyclin-dependent kinases in cell cycle, so the CDKs and cyclins. And then, do you want to take a break? Okay, let's take a break. 5 minutes, 10 minutes. Fourth is International HPV Awareness Day, and so that just because HPV causes all the troubles, not only cancer but also many other related diseases, and so I believe there are several. Wow, that's good. Let's take our meeting. Possible. I just want to let you know, so that beware, as a dentist, you may see in the future, you may have patients who have HPV infection or cavity, and so you have the ability to explain and what's going on, and so make sure your patients understand and aware of what they need to know about HPV infection. So now I just want to switch to head and neck cancer therapy. I'm not talking about like surgery, because the majority of those head and neck cancer can be removed by surgery, and the recovery is very good. So you can remove 99.9% of the cancer cells. The problem is for head and neck cancer patients, they're like, it's called a recurrence, or they have to come back with the same tumor. It's about, in 5 years, it's about 50%. In other words, half of the patients will have the tumor back after surgery or other therapies in 5 years. So that's not a good thing. And this is where we need a new therapy to save other 50% of the patients. And most of the time, the prognosis for the recurrence is very bad. Like 80% of them will die after, if they have a recurrence. So for cancer therapy specifically, here I'm talking about like solid tumors. I'm not talking about blood tumors. Surgery literally is the first choice, and also it's a standard of care, SOC, and so it's very effective to remove localized tumors, and you can get rid of 99.9% of the cancer cells, and followed by radiation therapy and chemotherapy. So they can be used for metastatic tumors. And however, the problem with radiation therapy and chemotherapy will be, they also affect the normal proliferating cells like bone marrow. Those are proliferating cells. And you still, again, you may have some radiation in the drug-resistant tumors. In most cases, because those tumors, they have higher content of, we call them cancer stem cells. So those cancer cells, they become like stem cells, so they are resistant to whatever therapy you put on them, and they have the potential to come back as a tumor. So then immunotherapy now is an option for people, even they are treated by radiation or chemotherapy. So there are many clinical trials are going on using immunotherapy to just make sure, just because the immunotherapy is more effective, more targeted. So then what's immunotherapy? So the first thing I just want to refer you back to the tumor again. So most of the time we don't think, just like I mentioned before, cancer is not a single disease. A vast of different disease related to uncontrolled cell proliferation. And also we believe cancer or a tumor, we can think about that. Yeah, we consider those cancer or tumor as an organ because it has its own blood vessels to provide blood and oxygen and nutrients to remove wastes. And also they have different supporting cells like endothelial cells, they have different stroma stem cells, and you have fibroblasts, and to have connective tissue to give them a shape of the tumor, just like an organ. And also we have some immune cells, for example, macrophages. Those cells, macrophages in the tumor, most of the time they are called tumor-associated macrophages. And also you have myeloid-derived suppressor cells, MDSCs, and also tumor-infiltrating lymphocytes, TILs for short, in the organ, in this tumor organ. And definitely you have a lot of cancer cells. So those components of the tumor, even those cancer become an invasive tumor, they still have those components, blood vessels, different fibroblasts, connective tissues, cancer cells, immune cells, and then they can form a new tumor in the metastatic site. And so even in this metastatic tumor microenvironment, it's very similar to the original, to the primary tumor. So this is the way for tumor, why the cancer is hard to treat first, and the second is they can recur. They can reoccur just because they can form a new tumor in other place. So there are, and I mentioned about, answer his question a while ago, and because those cells, the cancer cells are able to fold those immune cells. And then those immune cells don't think they are foreign, and they can inactivate immune cells by expression of inhibitory co-stimulatory receptors on immune cell membrane. For example, PD-1, program DEF1 is a receptor on T cells. And activation of PD-1 can cause cancer T cell inactivation and cause T cell exhaustion, so they don't have ability to be activated. And PD-L1 is a ligand to PD-1, and NEC3 is the other inhibitory co-stimulatory receptor on T cells. Myeloid-derived suppressor cells or myeloid cells, they become they become like suppressive to activation of T cells because they produce inhibitory cytokines to the T cells to inactivate them. And this is the work from my lab. We isolated blood cells from here. We used 45 head and neck cancer patient blood. So actually, right now we have over 100 of them. And also we isolate blood samples from 12 healthy normal people. And then we analyze the program DEF1 expressing T cells, and PD-L1 expression monocytes, and anyway, so you just by eyeball those data, you can see in head and neck cancer patients, they have higher levels of PD-1 expression T lymphocytes. They have higher levels of PD-L1 expression monocytes. All those are the immunosuppressive molecules and immunosuppressive immune cells in the circulation. So in other words, those patients, they have lower immunity. So their immune system is unable to recognize the tumor cells, even they know they are tumor cells, because they are exhausting immune cells in the tumor microenvironment. They are not going to kill those cancer cells. So so in my lab, we are trying to correct this to reduce like a PD-1 expressing T lymphocytes and other suppressive immune cells back down to the normal level. So then you have more activated T cells, and you have fewer inhibitory or suppressive myeloid cells. And then those T cells can recognize cancer cells and mount immune attack on those cancer cells, either limit their growth abilities or just kill them off. So that's the thought about immunotherapy. So there are different strategies for cancer immunotherapy, and then we can target immune inhibitory checkpoint blockade. It's called immune checkpoint blockade therapy. So I just mentioned about we have a lot of like PD-1 expression T cells. If you can inactivate the PD-1, then those T cells will be out of those exhausting state, become active. So this is why PD-1 expression T cells are unable to mount immune attack on cancer cells, just because activation of those. This is a PD-1. This is a receptor, and they can interact with the ligand from presenting cells or from cancer cells, can also produce the ligand called PD-L1. So the interaction of PD-1, the receptor, and PD-L1, the ligand, will activate the receptor. Activation of this receptor will inhibit activation of T cells from T receptors, from TCR, and induce exhausting of exhaust of those T cells, they are unable to activate cancer cells. And this is a cancer cell, they have high levels of PD-L1, PD-L2 expression, and they can inactivate the T cells. And also many other things involved, like macrophages, and so right now the PD-1 blockade therapy, there are many clinical trials, and we are talking about this is $5–10 billion market there, and therapy is by itself is really expensive. Like literally, we're talking about somewhere $10,000 to $20,000 a year. And so, however, there is a problem with this kind of PD-1 blockade therapy. And for metastatic and the recurrent head and neck cancer cells, the response rate for this kind of cancer to this treatment is only less than 20%. So in other words, 80% of those patients, they don't respond to it. For lung small cell or lung cancer, only 16% of response. Melanoma is very good, as we have like a 60% of response rate. And the renal cancer carcinoma, renal cell carcinoma is about 29%. So there are a lot of room to improve PD-1 blockade therapy that by different drugs, monoclonal antibody, or by using to analyze more mechanisms, what's going on there. And the other cancer immunotherapy is called a T-cell therapy. And theoretically, this is a non-specific because the reason it's non-specific. If we have a cancer patient, we can isolate a tumor, get a tumor out from the patient, and then we can isolate tumor infiltrating lymphocytes, those are T cells from the tumor. And then we grow those tumors in place and grow them up by adding IL-2. I mentioned about before, IL-2 is a cytokine, it's a growth factor for T cells. And then you have a lot of T cells. And then those T cells, you pick up the different, you grow the cells, those T cells in the place, they have a lot of wells. And then eventually you can pick up a clone of the T cells, they produce a lot of interferon gamma. So that's the indication, the activation of those T cells. Because Th2 T cells, Th1 T cells, they produce a lot of interferon gamma. And then we can produce tons of those, a lot of this kind of activated T cells by treating them with IL-2 and CD3. So proliferation and activation, and then put the T cells back to the same patient. And at the same time, we will irradiate the patient to kill off all the T cells, other T cells, only so the patient will have majority of activated T cells in the circulation. And since those patients already undergone surgery, so the tumor is supposed to be smaller, and then you have activated the T cells, and then the T cells will put the cancer cells on check and to inhibit their proliferation to reduce the recurrence and reduce the metastatic movement of those cancer cells. It has been used for metastatic melanoma patients, and it has been shown very successful. I'm talking about, say, like a response rate will be somewhere 25 to 40%. Not very high, but for this part of patients, that's very used. And the other T cell therapy is called CAR T cells. So CAR T cells, I think you have learned a lot about the T cells because Dr. Ellen Levan mentioned about the different types of T cells and activation of the T cells. So this is the concept is very simple. You have T cells, take them out, and engineer those T cells. So those T cells will produce antigen recognition side on all sides of the cells. And then inside the T cells, engineered cells, you have T cell activation signal peptides. And then eventually you can activate the T cells. So the idea is the antigen recognition antibody light chain on all sides of the cells can recognize the tumor, the antigens produced by the cancer cells. And then that will specifically activate those T cells. Then T cells can mount immune attack on those cancer cells to kill them. So one example here is the CAR T cell treatment for B cell lymphoma. Because B cell lymphoma overexpress a protein on the cell surface called CD19. And then you have T cells specifically recognize those CD19 as an antigen. So those T cells will be activated where the lymphoma cells are, and then mount immune attack on those cancer cells to kill them. So this one, in this case, the B cell lymphoma patients' response to CAR T cell treatment is about 90%. So that's really, really great news for this type of patients. And literally, you can cure all these patients. So here is the question. Just take a minute to read it. LEG3 is an inhibitory co-stimulatory T cell receptor of T cells to inhibit T cell activation. Yes, somebody? Because LEG3 is inhibitory. In other words, it will inactivate T cells. And for cancer therapy, immunotherapy, we want to activate the T cells. So if you have a monoclonal antibody against LEG3, and then you can make those T cells from like exhausting inactive form become active form. So they can mount immune attack on cancer cells. Right? So the answer is yes.
No, but if the problem is that the T cells aren't seeing the cancer cell because it's avoiding notice, what does it matter if you make more T cells?
So actually, those T cells will, so for the head and cancer, specifically for, we call it like oral cancer, those kind of cancer, those T cells will penetrate into or infiltrate into the tumor. You don't have a lot of this kind of T cells there, but they are there. They are in the surrounding region.
And they do recognize it as cancer.
Without any drugs, they don't recognize them. They are there, they don't recognize those cancer cells.
Okay.
So that's why but they are inactivated because PD-1 expression and LEG3 expression. So they are inactivated, they are exhausting. And however, if you use those antibodies to inhibit those inhibitory co-stimulatory receptors, and then those T cells in the tumor microenvironment will become activated. Once they are activated, they can mount immune attack to cancer cells. But the problem is, again, this is not a cancer cell specific, because literally you will activate all the immune cells in the body, the T cells in the body. But that's not a problem, because if you look at the data I just showed you, in head and cancer patients, they have much higher levels of PD-1 expression or LEG3 expression in cancer patients. So even you inactivate them, I mean, they can drop to the level just like healthy individuals. So that's not a concern that will cause all the different types of side effects. So I just want to summarize it. So actually, this is something I at the last minute I removed the slides because I thought about like vaccination is considered as immunotherapy. So like I think you will have lectures regarding HPV. There are HPV vaccines on the market, and those pharma companies really like promote their vaccine and ask the government to issue a policy change, and the schools can use that to vaccinate their kids. And there's a lot of like rejects from locals. I mean that's. However, for head and neck cancer, if even it's caused by HPV, then the vaccine has no use at all, because vaccine here is preventive. For head and neck cancer patients caused by HPV, actually they don't produce HPV virus at all. You don't see any HPV virus in the head and neck cancer patients with HPV infection. So they don't have the virus just because the oncogene from the virus already integrated into the genome of the host. So it's part of the host genome. There is no way that you can kill those. I mean, if you kill those virus infection, again, the tumor cancer cells will be still there. You have to figure out the other way to do that. And
actually, I thought that with the HPV vaccine, with regards to head and neck cancer, it was preventing you from ever getting those
that's right,
genes
preventing
into those
from
cells.
infection.
Right.
And you don't. So a lot of people are thinking about if all people get vaccinated, so at least 20% of the head and neck cancer can be prevented.
Mm-hmm.
But for those who already have HPV-associated cancer, then there's no value for use of the vaccine. And also I mentioned about inhibitor immune checkpoint blockade therapy. So those are like monoclonal antibodies against PD-1, and also against LAG3. There are at least 2 of them already on the market, and those big pharma companies are really they are working really hard to produce more this kind of antibodies. And just because of the single drug therapy, the response rate to this kind of immunotherapy is very positive. And also I mentioned about adaptive cell therapy, including CAR T cell therapy, and also just the T cells get activated, put them back into the patient for the therapy. So any questions? All right, thank you very much for your attention.